What's going on, folks? Thanks for joining me this evening. Nobody's cooked. You guys are going to be just fine on this test. It's exciting that we get to be first out of the gate. I'm really excited about that. And so my plan for this evening is to just work through some of these old FRQs. And what I mean by that is FRQs that represent previous exam administrations. So 2024 and 2025, and just go from there. That's my plan anyway. And if you guys have particular questions, like maybe one of the FRQ questions gives us an opportunity to ask other questions, and we'll do that too. Yeah, very happy to do it. Thank you for joining. This is fun. I like this sort of thing a lot. So I'm very thankful that my stream is not as crazy as my colleague's stream, who I went in earlier this evening, and the spam in the chat was unreal. So it was crazy. So thankfully, you guys are keeping it tame in here. Very, very, very nice. Thank you. All right. So we're gonna just start 2024 FRQ, and then do 2025, and kind of give you guys some tips along the way as well. The things that I've picked up over the years teaching. You're gonna be fine. You're gonna be fine. Yeah, Craig's stream was nuts. Hey, that dude is really good at what he does. So I've counted a blessing to be able to work with him. All right, let's do this. Okay, you struggle on natural selection? Well, we will be looking at natural selection quite a bit because it is featured in the FRQs quite a bit. So here's our FRQ. Actually, I can, since we started doing them digitally, that you're going to be taking your written or your multiple choice on a Chromebook or whatever, and then the FRQ is going to be on paper. And the booklet is literally a booklet that is just lines, and then there's one page that has a graph on it. The hardest thing is going to be at the top. At the top of the page, it'll say question 1, question 2, or whatever. So just make sure you're keeping up with that. Percentage for 5, off the top of my head, I think it's around 70%. Yes, you could need to know scatter plot and bar graph. You do not need to draw a best line of fit for error bars. You just draw the number that it tells you. All right. So yeah, and I'll actually show you how you can know how much your questions are worth as well. Yeah, the first 2 questions are the long questions. And we'll just look at the whole test just real quick. And these instructions are pretty helpful. They may not be current, but they're current enough. Questions 1 and 2 are long responses, about 25 minutes to answer each. And then 3 through 6 are about 10 minutes each. 3 through 6 have 4 points on them across the board. It's going to be 4 points. Questions 1 and 2 are going to have 8 to 10. The last few years, it's been divided evenly at 9 and 9, I'm pretty sure. So 18, and then 4 times, so 16, 35-ish points for the whole FRQ. Review Krebs, cellular respiration, yeah, I can do that. Yeah, I'm going to go through these FRQs, and as we have questions or as we have content come up, I will move over to those. All right, so let's look at this first FRQ together. Let's do this. So this is a Unit 5 question here. Crossing over in meiosis 1 is required for homologous chromosomes to properly align during metaphase and segregate during the first cell division. And so notice also questions will give you information, right? They will give you information that could be important. And so meiosis 1, aligning up metaphase, allowing them to properly, right, you get the idea. Describe the function of the S phase. So one helpful hint is that the first part of every question, part A of every question, is going to be just content, right? And we'll see this pattern. There's been a couple of exceptions to that, but in general, part A is just going to be a content question that could exist outside the context of the whole question. So describe the function of the S phase, and during the S phase, DNA is replicated. That's it. Don't write anything else. Anytime you see the word describe, by the way, and if you don't know this, everybody's has a different experience with this class, to say the least. And so when you see a bolded word like that, it's worth 1 point, unless it's the word construct, and then it's worth 3 points, right? Yeah, the exam is, what if I say the chromatid replicates? No, you would need to say that DNA replicates, because it's not chromatids until division starts. So, all right, some regions of the chromosome called hotspots display a higher frequency of crossing over than other regions do. Crossing over is suppressed in chromosomal regions near the centromere. The centromere region of a duplicated chromosome includes a collection of proteins that form a structure called a kinetochore. Scientists hypothesize that one or more of these kinetochore proteins are responsible for suppressing crossing over around the centromere. To investigate the hypothesis, scientists modified chromosome 8 in yeast such that in each cell, one chromosome from the pair of homologous chromosome 8s contained the gene encoding red fluorescent protein RFP, while the other chromosome from the pair contained the green. Okay, cells expressing RFP emit red light, and cells expressing GFP green light. Models of the modified chromosome 8 are shown. Okay, so we have the red, and then so this is a homologous pair, right? And one of the pair has red, the other of the pair has green. And the idea is that you're trying to see if crossing over happens, right? So here's our picture. This is after crossing over, right? After crossing over, this part switches spots, and now you have red and green on a chromosome together. Okay. So we answered A, part A, part 2, whatever. See how this is A1 and A2. Explain why some haploid cells form after meiosis in this experiment will only have one fluorescent marker. Okay. The reason that haploid cells after this experiment will only have one fluorescent marker is because crossing over is going to occur, and sister chromatids separate during meiosis or meiosis 2, and so that is why. Okay, because remember, homologous pairs separate, and so this one will separate from this one, and then at the end, these two pieces are going to separate, and these two pieces are going to separate. And so if one of them gets this one and one of them gets that one, then only one of those cells is going to have a fluorescent marker in it. Okay, good. And there's more, just when you thought Wow, this is a big question. 2024 was a hard exam. Scientists then investigated whether attaching individual kinetochore proteins to a specific DNA sequence present in a known crossing over hotspot on chromosome 8 affected the frequency of crossing over at this location. In their first experiment, they examined 3 groups of yeast cells containing the modified chromosome 8. Group 1 contained no kinetochore proteins attached to the hotspot. Group 2 contained the kinetochore protein CTF attached to the hotspot. And group 3 contained the kinetochore protein IML attached to the hotspot. And again, the hotspot is it's described up here, right? And so hotspots that display a higher frequency of crossing over. So don't forget that as you're coming down here. What I tell my students is it's good to annotate. It's good to maybe make little marks. Just know that if you write anything in your test booklet, it will be graded unless you strike through it or if it's not in the lines. For each group, the scientists determining the frequency of crossing over between RFP and GFP genes. To determine the frequency, the scientists added the number of cells emitted, both red and green light, to the number of cells that emitted no light and divided the total number of cells. And there you go. So here's our frequency of crossing over. If there aren't any hotspots, right? Let's see, no kinetochore proteins attached to the hotspot. And so this one has no proteins at the hotspot. And remember, the kinetochore proteins are going to prohibit crossing over. CFT, and then IML. Yeah, there is a timer on your digital screen. Good. All right. Identify the control group for the scientist's first experiment in Figure 2. All right, in Figure 2, we have different kinds of treatments here, right? And so first of all, I don't want to assume things. When we're looking at a graph, the dependent variable is on the y-axis and the independent variable is on the x-axis. The independent variable is what's being manipulated. The dependent variable is what's being measured. And so the way to find a control is to ask yourself what is not manipulated here, right? And so this is not being manipulated. So identify the control group. It would be the group that has no proteins, no kinetochore proteins at the hotspot. All right. In a follow-up experiment, scientists created a modified CTF in which the DNA binding portion had been removed. They compared the frequency of crossing over yeast cells in the presence of absence of unmodified with the yeast cells in the presence of absence of the modified. In a follow-up experiment, justify why scientists used a modified protein that is unable to bind to DNA as control. All right, so again, this is just basically what is the definition of a control. The reason that they used this one that is this modified that is unable to bind, so that they would have a basis of comparison for the one that is able to bind. And identify, do I have any ways to de-stress before the test? Yeah, the best way to de-stress is to know that in 20 years you might not remember if you passed this exam or not. That's how it's important, but it's not that important. Yeah. And that's a lifelong coping pattern, by the way, being able to determine which things are important and which things aren't. And so developing that lifelong pattern will be more important than what you make on this test. So, yeah, you can major in biology without a 5 I majored in biology, and I didn't even take AP Bio. So there you go. Yep. Identify the independent variable in the follow-up experiment. It is what is being manipulated. And so we just find what's being manipulated, and it is the modification of the CTF protein, right? The reason that you take this class if you're going to law school is because it's good to do hard things. Part C. Based on figure 2, describe the effect on the frequency of crossing over when CTF is attached to chromosome hotspot with the effect of IML. Okay, good. So this is asking you to describe the difference between these two, and you can just say there is a difference between them. Or you can say IML has a higher crossover frequency than CTF. Don't get crazy on this one, right? It's just asking you to describe the effect, and that's all. You're looking at this graph. If these error bars were overlapping, then you would say there was no difference, right? Yeah, I mean, the exam is stressful, but it's not, though. It's not. You're in here, like, getting help, so that's good. It's just not that stressful. It's not, I promise. I take one every year. Well, I don't, but you get the idea. It's just not that bad. You guys will be fine. You got to have confidence. You've been studying for this all year. All right, part D. Predict the effect on the number of copies of chromosome 8 likely to be present in the resulting daughter cells when CTF is attached to the hotspot. So predict the effect on the number if CTF is attached to the hotspot. CTF is not going to affect that, and I mean it's not going to cause non-disjunction or anything, and so it's just going to be 4. This is not going to have any effect. So I went to college at Murray State University. All right. Explain how the presence of hotspots could increase the likelihood that a population will survive in the presence of selection pressures. Okay, good. And so someone was asking about natural selection earlier, and this is important right here, right? Someone was asking about natural selection, and they're struggling with that. So look, explain how the presence of these hotspots, which causes more crossing over, right, will increase the likelihood that they will survive. What does crossing over do to a population? It increases genetic diversity, right? And genetic diversity is going to increase fitness. And so that's the way you need to think about it, right? So, yeah, and you guys are talking about keywords. So let's just look at this question real quick and just sort of overview. And just truth be told, like the chat's kind of moving fast. And so if you have particular questions, that's fine. But I'm just kind of looking over there every once in a while, just so you guys know. So you have 1 point here with describe, right? You have 2 points here with identify. Let me mark this out. B3. Oh, yeah. No, I got B3. I just did that one with the if it increases genetic diversity. Predict and justify, I did that at the same time. Predict the effect on the number of copies of chromosome 8 likely to be present in the resulting daughter cells when CTF is attached to the hotspot. CTF has a lower frequency of crossing over, but it doesn't prevent, crossing over is not going to change the number of chromosomes, and so predict the number of copies of chromosome 8. It's still going to be 1 copy because CTF does not cause non-disjunction. That's my thought on that, and that's my justification. So let's see, 1, 2, 3, 4, 5. With identify as well, you do not need to write whole sentences. You just need to write the thing. The ampicillin lab. If we're able to get to that, I will. The lab probably won't be as big of a thing. Yeah. Just questions about how that works, and I think there's one on one of these FRQs has something like that. So 1, 2, 3, 4, 5, 6, 7, 8. Did I get that right? Yeah, it's only 8 points there. No, yeah, I'm glad you mentioned the restating. Do not restate the question. Don't. Just write the answer to it. Never restate the question. This is a science class. This isn't 5th grade middle school English. Don't. And that's, if you're one of my students, then you've heard my rant about that. No, don't restate the question. So for this one, identify the independent variable. For the treatment, I would say the modification of CTF, because it's just identify. Just simply write your answer and then walk away. If you rewrite all of that, the graders are not reading that. They're not. Trust me, they're not reading it. So you do have the right and complete sentences, but you don't have to restate the question in order to craft a complete sentence. That is something that you guys do not have to do. It is. I think it's good for elementary and middle school, but you guys are hammered that so much that by the time you get to high school, you have a hard time letting go. No, and not paragraphs. You could answer this question in a lot less than this thing gives you so much paper. But let's see. I'll just show you. Here I have a scoring guide here. Here's a scoring guide, right? And this is the question that we just answered. Notice how they're answering these questions, right? Describe the function of S phase, replicate DNA, right? That's it. Don't. The function of S phase is to replicate DNA. That's it. And notice they underline these things because they're looking for those keywords, right? And so don't write more than, and these bullets, these are different kinds of the answers that they're looking for, right? Let's look at this one here's this one. Hotspots would increase genetic diversity, therefore they would have more ability, they would have more fitness, right? That's it. It's just, you don't have to, and so they said it would be more likely that individuals would survive and reproduce. You could just say it would increase individual fitness. You could even save some words there if you know your vocab. So, all right, let's move on to this question. So here's the word describe here, right? To investigate how increases in environmental temperatures affect the metabolism of certain organisms, researchers incubated liver cells from toads. There's all kinds of weird crap happening in these FRQs. Researchers incubated liver cells from toads at different temperatures and measured 2 markers of metabolic activity, the rate of oxygen consumption and the rate of ATP synthesis. Okay, so this is, some of you guys were asking about Krebs and that kind of stuff earlier, and so here is, here's our question. This is looks like a Unit 3 question. So let's do this. Effective metabolism. Okay. So again, here's part A. Describe the role of water in hydrolysis of ATP. Remember, part A is always going to be just simply content-based rather than, you don't need the whole prompt, right? So it's just why it's up here. Yes, there are 6, I bet someone in the chat can probably answer your question there. There we go. Thank you. That's better than me doing that. All right. So describe the role of hydrolysis of ATP, of water. So remember, the word hydrolysis means adding water to break, right? We could come up here to our Unit 1. Let's see here. let's look at hydrolysis. So here's hydrolysis, right? Let's look at a different picture here. I don't have a hydrolysis of ATP picture, but it doesn't matter. It's all the same. So here's a polymer, right? And you add water to it, and it breaks the covalent bond between those two monomers. And so going back here, what is the role of water in the hydrolysis of ATP? Water is added to break the third phosphate from the other two. Something along those lines will be sufficient. Let's see. I'll consult the scoring guide so you can kind of see. Water's added to split ATP, to split a phosphate from ATP. Water breaks down ATP. Literally, look what they would have accepted here. 3 words. And so there's no reason for you to write paragraphs on this stuff. If you write paragraphs, they're not great. They're not looking at the whole thing. So, all right, here's our table. Using the template for your response, construct a bar graph. This one even tells you the type of graph to construct, which is good. If it didn't tell you what to construct, how could we know that it's a bar graph? The way that we could know it's a bar graph is because we're comparing two things. We're comparing the rate of oxygen consumption and the rate of ATP synthesis, right, at these 3 different temperatures. Okay. I'm interested to see how they did this. What are they? They're measured 2 markers, and so this is what's being measured, right? It tells us that we're measuring the rate of this and the rate of this. So this is our y-axis here, right? This is going to be our y-axis, and this is going to be our x-axis. You don't get a choice on that, by the way. You do not get a choice between which axis you want to put it on. What's being measured is on the y-axis, and what's being manipulated or changed is on the x-axis every time. And you can see the way that they graphed it here. They actually accepted both. Oh, no, I see what they did. No, they didn't accept both. We have rate over here, right? And then you have your different temperatures. Here they put them in groups of oxygen and ATP, and over here they put them in groups of 20, 25, and 30. And then, so either one of those would have been fine. This is like a modified bar graph, right? If I was doing this, I would just use the table, and my X would be 20, 25, and 30, and I would have a bar for, or 2 bars for 20, 2 bars for 25, and 2 bars for 30, and that's the way I would do it, just because of the way the table's organized. It does matter what type of graph you do. For this one in particular, since it told you to do a bar graph, that would definitely matter. If you drew anything else, it would be wrong. And the word construct, by the way, it is the only thing that varies from our normal pattern. The word construct is worth 3 points, and we can go and look at those 3 points actually. Let's go look at them. You have to have the data represented correctly, right? You have to label it appropriately, and you have to have the error bars correctly plotted. Yeah, we practiced all kinds of types in my class, so, but I think they default to bar graph just because it's just helping kids out, but I don't know. We practiced a lot of graphs in my class. Let's see, based on the data provided, determine the temperature. Yeah, you need to title the graph, for sure. Theirs aren't titled, but I think the axes, as long as the axes are labeled, you're probably fine. So, Unit 6 review. Yeah, I have a video reviewing Unit 6, and you can watch that and whatever content we get to here. Yeah, So whatever content we see here, we'll review directly if you have specific questions when we get to that content. But yeah, I have a Unit 6 video for sure. Yeah, and the error bars, don't do anything crazy here. Just literally count up 2.2 and 2. Don't double this. All right, I've seen more teachers tell their students to double this because of this. Don't. Yes, you always have to put error bars. If you don't put error bars, it's wrong. You put error bars if it's a scatter plot, if it's a line graph, or if it's a bar graph. It doesn't matter. You're going to put those. See, they didn't double. Just don't double. Don't double. And determine, based on the data, determine the temperature in Celsius at which the rate of oxygen consumption is different from the rate of oxygen consumption at 25 degrees. And so we're going to come up here. I'm just going to look at their graph. So oxygen consumption at 25, which one of these is different? It looks like both of them are going to be different. And let's just go look at their graph. Oxygen consumption at 25 is this bar. 20 is not different. Notice 20 is actually not different than that. Interesting. But 30 is different. And how do I know it's not different? It's because the error bars overlap. And it should say that for us. And notice here, it says determine the temperature, 30. Now, I would count theirs wrong because they didn't put the unit. And so make sure that you put unit every time. 30 is not just a random number, but it has to, it has some kind of unit behind it. So always put your units. Always put your units. Even if it's like giraffes, 30 giraffes, which I don't know what kind of question would have you write that answer, but you never know. Okay. Based on data in Table 1, describe the effect of temperature on the rate of ATP synthesis in liver cells and toads. Okay, so describe the effect of temperature and ATP synthesis. It causes it to increase. That's it. Don't write anything else in that, right? Look, it's going to be that simple. As temperature increases, the rate of ATP synthesis increases. Don't get cute. It doesn't want you to be cute. Just tell them what's going on. Oh, this is a good one. I like this question. Based on the data in Table 1, calculate the average amount of oxygen consumed in nanomoles for 10 milligrams of mitochondrial protein at 10 minutes. No, you don't have to show your work. I will demonstrate here. So at 25 degrees Celsius, oxygen consumed is 16.5, right? And this is per minute per milligram. You don't need to know any of them because you get a formula sheet, but I would probably be versed in Hardy-Weinberg and chi-square. I don't think I've seen a water potential problem. I haven't seen a problem, but I've seen one of the practice exams had you determine the difference between two water potentials, like which way water would go. And so let's look at this question. If this is 16.5 per minute per milligram, what if it was 10 milligrams in 10 minutes? Then you would just multiply this number times 10 times 10 or times 100. And so that'd be 1650 nanomoles is what that would be. And so you wouldn't have to show that. You would just write 1650 nanomoles of oxygen. Do you need to put oxygen? No. So, yeah. Yeah, you're going to have a test booklet that's going to have lined FRQ paper on there. Part D, oligomycin is compound. For the graph question, you will have, you know what? I can do this real quick for you guys. Because I feel bad for you. And it could be that your teacher's showing you this and you just don't know. but I'm feeling a little bad that you guys are struggling with this. So let's see here. We're going to add a page, and we're going to do that. Okay, so this is our sample booklet. This is your test booklet. All right, this will hopefully put some of you guys at ease. I've gotten a lot of questions about the test booklet this evening. So your multiple choice are going to be digital, right? They're going to be on a Chromebook or whatever. But for your FRQ, you're going to be given this test booklet. Now, your questions will be on your Chromebook, but your responses are going to be recorded in this booklet. And so you'll write something there, and then look, it's literally just blank paper, right? I mean, you can go and watch my videos. Just go watch the review videos tonight. Just the Unit 1, Unit 2, Unit 3 videos. So look, begin question 1. It says do not skip lines. If you skip lines, they're going to grade it still, but I don't know why you would. This is not double space. And it's asking you to use a pen. The reason it's asking you to use a pen is because they're going to get a scanned version. Do not write your name. Do not write outside the box because it's not going to be scanned. If you write outside the box, it's not going to be graded. Notice this is still question 1. Still question 1. They give you way too much. You don't need all this. You could answer all of question 1 on this one page here. And then here's your graph, right? This is the graph they're going to give you for question 2. See, I have a nice graph here. There you go. And then I have some other question 2. Here's question 3. There's 4. Notice on these short ones, you just get one page, and then that's it. After the exam, there's some instructions for you. There you go. So hopefully that calms some of your nerves about the about that. Yeah. All right. Let's go to part D. Oligomycin. Oligomycin is a compound that can block the channel protein function of ATP synthase. Predict the effects of using oligomycin on proton gradient across the intermitochondrial membrane. All right, so we're gonna go find a picture of this. Here we go. This is a great picture. All right, so here's cellular respiration, right? Well, we don't have glycolysis in here, but we remember that there's a proton gradient building up in here, right? And if building up in the intermembrane space, and if oligomycin blocks the protein channel function of ATP synthase, then what's going to be the effect of the proton gradient? Well, the gradient is never going, it's just going to keep building up. There's never going to be a release, and there's never going to be the production of ATP, right? So that, and justify my response. Well, so what would be the effect on the proton gradient? It's going to build up. It's going to continue to build up. Justify, because when protons pass through ATP synthase, it causes it to make ATP. There's going to be something like that. Increase, becomes steeper, is what they said, right? And without the protons, more protons will accumulate. Anything like that we were saying would be fine. And let's just review how many points was this one worth. 1, 2, 3, 4, 5, 6, 7, 8, 9. So this one was worth 9 points. Okay. Sweet. On to the short ones. You don't need to memorize any of those formulas, thankfully. You're given a sheet that looks like this, and you don't need to memorize any of them. It's kind of nice. Yeah, percent change. I think of percent change as like logic, and so I don't think of it as a formula. But And then for Hardy-Weinberg as well, the one thing that you're not given here, you're not given that big A is equal to P squared, and big A little a is equal to 2PQ. God, this is disgusting. And the little a is equal to Q squared. You're not given that information, but everything else you're just given. 2024, back to the FRQs. All right, so let's look at this short one. So this is a short one. This one should take you about 10 minutes to answer. Notice there's 1, 2, 3, 4 points, right? Okay. I've not seen a single Simpsons or population math on an AP test question yet. So, and my students have confirmed over the years that there haven't been those questions. But this will be the year. All right, question 3. To investigate whether red blood cells of animals lose the ability to take in glucose from their environment as they age, scientists collected red blood cells from guinea pigs that ranged from 1 day old to 7 months old. Scientists incubated the equal number of red blood cells in separate culture dishes that contained a 300 nanometer solution of radioactive labeled glucose. The amount of radioactively labeled glucose present inside red blood cells for each group was measured over time. And so basically what's going on here, do red blood cells use the ability to take glucose from the environment as they age? And they took red blood cells from different things and added glucose to them. All right. And again, here we have part A. Describe the difference between passive and active transport. Has nothing to do with the prompt, right? It's just a straight content question. Describe the difference between passive and active transport. Passive transport does not require energy. Active transport does. That's it. Don't write anything else. Justify why scientists use an equal number of red blood cells in each culture dish. Okay, this is a great time to tell you this. So questions 1, 3, and 6 are going to have like scientific method type questions on them, right? Science practices, stuff like that on there. Experiment design. And so this is an experiment design question. Why do you use an equal number of red blood cells? So that there's not two variables, right? If you had a different number of blood cells, then your variables would be age and number of blood cells. But because everything's the same, the only variable is age. Okay. Part C, glucose transporters are required for facilitated diffusion of glucose into red blood cells. Okay. The scientists claim that the expression of gene encoding these transporters decreases as guinea pigs age. If the scientists' claim is supported by experimental data, predict the effect of increased age on the amount of radioactive labeled glucose present inside the cells of each group. Okay, so if glucose is less able to get across the membrane because of decreasing transporters over age, then there's going to be less radioactive glucose inside the cell as they age. My justification is because age affects the amount of glucose able to go into the cell. Pretty simple. Krebs and the Calvin cycle are not the same thing. So they're opposites of each other, actually. And one is happening in the mitochondria and the other's happening in the chloroplast. I'm actually curious how they answered this one apart. Yeah, fewer transporters. It's just that simple. It's that simple. All right. Question 4. Now again, 4 points. 1, 2, 3, 4. The common wild oat is native to regions in Europe and Asia, but is an invasive species in Central California grasslands. This is an ecology question. In California, the common wild oat has almost completely replaced some species of native bunchgrass. Researchers found that aphids, a type of small insect that often carries plant viruses, have a much higher reproductive rate in grasslands that include the common wild oat in grasslands composed of only native bunchgrasses. Additionally, the viruses carried by the aphids appear to affect only native bunchgrasses and not the common wild oat. Native bunchgrasses infected by viruses have a higher death rate than do native bunchgrasses that are not infected. Okay, so to summarize what's going on here, you have the wild oat that's came in, which increases the amount of aphids, and the amount of aphids is destroying the native grasses. And here's part A again. Not going to be, it's going to be completely out of context. Describe the change in resilience of an ecosystem when there's a decrease in the number of species. As the number of species decrease, the amount of resilience decreases. It's not asking you why that happens. It's just asking you to describe the change in resilience. It goes down, right? I'll probably stream till around 9 or so. I'm going to stream until I finish 2025 FRQs. That's when I'm going to finish. I may ask a couple of content questions, maybe go over a couple of things that are like hot button stuff. Yeah, I tell you what, after I finish 2025, stick around for 2025, and I will go over all of photosynthesis and cellular respiration with you guys. How about that? I'll go over the whole thing, top to bottom. All right, we'll do that. Part B, explain how the addition of the common wild oat affects the number of native bunchgrasses that can be supported by the California grassland system. Explain how the addition of the common wild oat increase the number of parasites and disease, and so that is going to directly affect the California bunchgrass. Researchers suggest adding ladybugs, a predator of aphids, to California grasslands. Predict the effect of adding ladybugs on the abundance of native bunchgrass population. If you add ladybugs, it's going to decrease aphids, which is going to decrease disease, which is going to increase bunchgrass. Right? And so predict it's going to increase. Why? Because you are decreasing the amount of disease in that population. Very simple. Moving on to part 5. Researchers study mechanisms that enable or prevent speciation. Oh, somebody was asking about post-zygotic earlier, so here we are. Researchers study mechanisms that prevent or enable speciation. Describe a post-zygotic mechanism that prevents gene flow. So there were 3 post-zygotic mechanisms. Let's go ahead and let's go ahead and find that here. Here's the 3 post-zygotic mechanisms. This is just a straight vocab question, right? And let's say that you knew this one about the mule, right? And you're like, I don't remember the name of it. I just know that if 2 species have a baby, the hybrid is sterile. Then just say that. Just say what you just said. When a horse and a donkey have a baby, the mule is sterile, right? And then that would be good, right? So something like that. All right. We got to read this stuff. New genes can evolve from non-coding regions of the DNA. It is not until certain regulatory elements are present in the DNA that non-coding regions becomes a new functional gene that encodes a protein. These regulatory elements include a promoter, 5' untranslated region, UTR for short, followed by a start codon, 3' UTR. And got the thing. All right. Wow, this is unreal for a short one. This is crazy. Good times. All right. Researchers studied the evolution of the family of antifreeze glycoprotein encodes genes in Gattidae, a family of marine fish known as cods. When present in the fish, these glycoproteins reduce the freezing temperature of fish. Researchers compared genomic sequences in 9 cod species and 1 non-cod species. They recorded the presence and the absence of functional AG genes as well as AG-like sequences that are similar to functional gene but have undergone mutation. Do not. Okay, lots of stuff here. How to split your time. The instructions will, I literally tell you. Questions 1 and 2, about 25 minutes, and questions 3 through 6, about 10 minutes each. So that is a way to think about it. All right. Part B, based on the data in Figure 2, explain how changes in the genome enable cods to survive and reproduce after a period of freezing temperatures between 10 to 15 billion years ago. All right. And how do they survive this period of freezing? It's because they have this glycoprotein that reduces their freezing temperature. So, yep, that's it. I don't. That one can't be much harder than that, right? Yeah, emergence of. Right, these genes that prevent freezing. Right, good. Okay. Using the template in the space provided for your response, place an X on the phylogenetic tree to represent the origin of the function of the AG gene. Let's see. The Gattidae, which is going to be right here. And so we're going to put that around this freezing point here. Is what we're going to do. Because notice this is where it's going to split off, and so we're going to put that here. That X is going to be right there. Yeah, I think that you mentioned that AP Chem is harder, or AP Bio is harder. For some people, it is. Chem was a lot harder for me. So, based in Figure 2, explain how the genetic differences among species determine the habitats in which they can survive. Genetic differences are going to determine phenotypes, and phenotypes are acted on by the environment, right? And so that's going to be the thing there. Genetic differences are going to produce different phenotypes, and different phenotypes are going to be able to survive in different environments. So that's it. Question 6, finally at the end of this one. Scientists can quantify the state of translation as ribosomes move along an mRNA from one codon to the next. Using a procedure called ribosome profiling, scientists measured how long a ribosome remains stationary at each codon of messenger RNA. Oh, I remember this question. They determined the average translation rate across all codons is 5.2 amino acids per second. But that average translation rate for specific codons in different RNA sequences can vary widely. These variations in translation rates are thought to facilitate correct folding of the protein being produced. The rate at which 3 different codons was translated measured 100 different messenger RNAs. The scientists determined that the distribution rate, number of times each was recorded for each of the 3 codons, and there you go. And so this is the number of times rate was recorded, and this is how long it took. And so towards this end is slower, right? This is slow. This is faster. And so GAG is on average fast. AUU seems to be really fast, right? And UGG seems to be slower than those two. All right, let's go look. Using the data in graph A, identify the rate that was recorded for the greatest number of times for GAC. The rate at the greatest number of times was 150 milliseconds per codon, right? Right, was it. Just literally, can you read a graph? Using the data in figure 1, graphs B and C describe the variation in translation rate of AUU compared with UGG. AUU is translated faster than UGG, right? Because it has faster, takes less time. Is another way to get to say it. Scientists hypothesize that tRNA molecules that bind to UGG proteins are available in lower abundance than tRNA for AUU. Support the hypothesis. Support the scientist's hypothesis is probably going to be replaced by the word evaluate, which is fine. Either one is fine. How would you support that? It's because it takes, if there's less tRNA, it's going to take more time to translate that, and this makes sense. The data shows that. Amino acids can be encoded by multiple codons. In many organisms, certain codons for the same amino acid occur more frequently in RNA than do other codons. Based on the data provided, explain why the use of one codon over another for the same amino acid might result in increased levels of protein production from a particular. Explain why the use of one codon over another. Because one codon would be faster, right? Because this shows that the rate is going to be faster for some codons than other codons. And so it's going to produce more amino acids because some codons are faster than others. Something along those lines. Let's see on this one. I'm actually curious how it's been a while since I've looked at this. So certain codons are faster. So it's just literally, it's as easy as you think it is. Don't overthink these things at all. Okay, so we're going to start 2025 here in like just a minute. If you will let me just take a quick break, I'd appreciate that, and then I will be right back. Okay, here we are. Thank you guys for allowing that break. Really appreciate that. Here we are, 2025 free response questions. Let's do this. You can write bullets, but you have to write in complete sentences. So, actually, you know what? let's look at these instructions together, shall we? Let's look at these. Let's look. Section 2 is 6 questions. This is FRQ. This is last year's test, so these instructions are pretty well current. So let's look at this together. Read each question carefully and completely. Answers must be written out in paragraph form. So there you go. This says bulleted is not acceptable, so I was wrong. I thought you could use bullets if they were complete sentences. Paragraph form. Do not spend time restating questions or providing more than the number of examples than called for. For instance, if a question calls for 2 examples, you can earn credit for the 2. That's it. Don't write more. Labeled diagrams, available paper, calculator, pace yourself. Okay, and then it tells you this. So yeah, paragraph form, and when I say paragraph form, just write sentences, right? I would not use gel because gel are less reliable. So. All right, let's look at this. Let's look at this FRQ. All right, this one's gonna be fun. All right. Most proteins that are secreted from cell must be transported to the endoplasmic reticulum during translation or after translation. Part A is, let's see, describe the function of ribosomes. The function of ribosomes is protein synthesis, right? Good. The site of protein synthesis. And so, how would you write that answer? How are you formatting this? Ribosomes synthesize proteins, period. Subject, verb, direct object. Not whole paragraphs. That's just, look, this is what the graders are going to get, and this is the correct answer. It's 3 words. Don't do anything crazy. Right? Don't do anything crazy. Just don't write paragraphs. If you write a story, nobody's going to read it. Trust me. All right. For proteins transported during translation, this process begins in the cytosol, pauses when a specific sequence of amino acids are translated. The translation complex is then transported to the surface of the ER when translation continues. Proteins that are transported after translation are translated entirely in the cytosol and transported to the ER. In both instances, translated proteins enter the ER through a protein channel in the membrane of the ER. Researchers studying 2 types of protein transport identified the ER membrane, protein SR, is necessary for transporting during translation, while the ER membrane SEC62 is necessary for transport after translation. To investigate which transport mechanisms is used for different proteins, researchers first created small interfering RNAs that reduce the expression of either SR or SE62. They treated groups of cells with either SR, siRNA. God. These abbreviations are there to make it harder on purpose, right? Or SEC siRNA to determine the relative amount of SR protein in each group of the thing. And so siRNAs reduce expression, right? And so that's the idea. And here's our thing. We have our controls, and we have these. Identify the dependent variable in experiment. Dependent variable is always the y-axis, and you do not have to write a sentence for identify. So identify is the relative protein content. That's it. Don't write anything else. Justify why researchers include the control of measuring relative amounts of both SEC62 and SR proteins in the cell that were treated with SE62 only. The reason that you do this again, why we have a control, so that we can have a basis of comparison. That's it. Their answer is going to be something like that, right? It's going to be to determine, to determine whether SEC62 is affected by both. So it's just a basis for comparison, right? To determine by that protein only. And so it's just a way of saying, does this only affect this one, or does it affect both of them? It's just a basis for comparison. Part 3. So there's 3 points so far. 1, 2, 3. Part 3. Based on Figure 1, describe the effect on the production of SR proteins when cells are treated with SEC62 sRNA. What is the effect? It increases the effect. And so how do I know? I actually got this one wrong when I was doing this on my video. So I'm comparing it to the control, right? Here's the control, and when I treat it with this, it's higher than the control. So to describe the effect, it increases the production. That's it. More graphs. The researchers then measured the amount of 3 different proteins that was transported to the ER cells treated with those things. The researcher calculated the percent transported relative to cells treated with control siRNA. And here they are. Identify the independent variable in the researcher's second experiment. The independent variable is going to be the siRNA added to the cells, right? Just the x-axis here. So, boop. Identify the proteins that when treated with SC62 siRNA showed an increase in percent transport compared to the control. So an increase in this. So this one was decrease. This one was increase. And this is, so proteins 2 and 3. Protein 1 is encoded by 234 nucleotides, while protein 2 is encoded by 495 nucleotides. Assuming all nucleotides for both proteins encode amino acids, calculate the difference in the number of amino acids between the 2 proteins. So this is key here, so we don't have to deal with a stop codon, all nucleotides. And so 234 nucleotides is going to encode for, let's see, 78. And this one's going to code for 165. And the difference between 165 and 78 is 87. Right? So that was pretty simple. And if I'm going too fast, let me know. I'm noticing that chat is talking about not AP biology. But anyway, it's just fine with me. Researchers claim that protein 1 is only tested protein that is transported to the ER following the complete translation in the cytosol, using data to support the researchers' claim. Let's see. Transported to the ER following the complete translation in the cytosol. I'm curious what they wrote on this one. Yeah, only protein 1 showed a reduced percentage of transport when cells were treated with that. Right. Okay. And that's just because that RNA is going to prevent translation. See, these two both increased here, and this one decreases, and so when it was translated in the cytosol. For any protein that enters the ER, researchers claim that amino acids close enough to the protein's amino terminus determine how likely the protein is to pass through the protein channel. Justify the researchers' claim based on your understanding of factors that affect the transport proteins across the membranes. How close the amino terminus, how likely it is to pass through. It's because the amino terminus has a charge on it, and it's not going to be able to pass through the membranes without a transport protein. That's it. Moving on to question 2. Many insects rely on pheromones, chemical signals that are released by females to find mating partners. Scientists hypothesize that in a certain type of moth, the behavior of male moths is in response to the pheromones is regulated by extracellular signaling. Many receptors are embedded in the plasma membrane. Describe the polarity of the portion of the receptor that is inside the membrane. If it's inside the membrane, it's got to be nonpolar because the hydrophobic tails are nonpolar also, right? And so that would be simple. Again, part A is always going to be outside of the context. Let's look at this graph. How do we know which graph to use? Well, this is going to be a bar graph. I can already tell because you just have comparisons here. You have moths injected with saline versus moths injected with siRNAs, and general versus oriented activity. And this is a long question. It's all right. Using the template, construct an appropriate type of graph. This is an appropriate type of graph. And so the way I would do this is I would put this on my x-axis because being treated with saline is a control versus the presence of siRNA. And then I would have one set of bars that were general activity and the other set of bars that were oriented activity. You can probably see how they did it here. Yeah, this is how I would have done it. But notice if you flipped the X versus the different kind of bars, it's fine. But the Y-axis is always the same, what's being measured, right? So never change that. Determine the type of activity that was affected by inhibiting the expression of the DOP receptor. Let's see, we didn't read this part, did we? To investigate whether binding of 20E receptor, or the binding of 20E to its receptor, DOP-E, affects the behavior of moth injected with saline or interfering that inhibit the expression of gene encoding DOP-ERC. These scientists then expose the moths to the pheromone to determine the effect. Okay. And so which one is affected by that? Yeah, it's just literally, we can look at this graph and see. This is the general, right, versus oriented. And you can see that the activity of the moth goes way down on the ones that had the siRNA injection for the oriented one. And so that's the way I would look at it. SIR, and you could do it this way too, right? And you can see how they write this. Oriented activity was affected. Very simple, right? Based on data table 1, identify the treatment group in which the oriented activity was greater than 50% of the general activity. Was greater than. Identify the treatment group in which the oriented activity was greater than 50% of the general activity. Both saline and yeah. Okay. The scientists studied some moths with a mutation in the gene encoding the G protein. The mutation prevents GTP from displacing the GDP bound to the G protein. Based on Figure 1, predict the effect of this mutation on the oriented activity in male moths exposed to the pheromone. They would decrease, right? Because the G protein is going to be that in-between protein, that relay protein, and so it's going to decrease their activity. Expression of the gene encoding DOP-ECR is low in both male moths during their first few days as adults when they are sexually immature. Gene expression rapidly increases as moths reach sexual maturity. Scientists claim that this increase in gene expression increases the likelihood of males finding females with whom to mate. Use the evidence from the information provided to support the scientist's claim. Is low. Scientists claim the increase in gene expression increases the likelihood, right? Because gene expression is low before they reach sexual maturity and increases at sexual maturity, and gene expression rapidly increases in moths as they reach sexual maturity, and the scientists claim that the increase in gene expression increases the likelihood, and right, evidence is as they increase sexual maturity, they're going to have increased gene expression. That one almost seems odd, so that was kind of strange. I'm actually curious what they wrote. I may be off on this one. No, increase rate, increase orientation. Right, it's just that simple. Based on Figure 1, explain how an inhibitor of the DOP-ERC pathway might serve as an effective chemical to protect crops from moth damage. If you're able to inhibit that, then you're able to keep them from having that reproductive activity, and there's going to be less moths. The different guys for the test. No, you don't need to know any of those people. They're not on our course description. I think it's a good. If you guys are just as a hint here, do you need to know the different scientists? No. And so just to kind of explain, if you take another AP class, my strong recommendation to you would be to download the course description of the class, and it shows you everything that's going to be on the test, right? It's a lot of information, but you can see on there that you don't have to know any of the scientists' names. This is not a trivia class, right? And so you're not going to have to be able to list. Anytime that there's a scientist named, it usually tells you what they do. Yeah, just knowing trivia is not really very helpful for this test, right? Understanding ideas is more important than that. All right, let's look at number 3. Buffalo grass is an invasive grass species in the southwestern desert ecosystems. It's threatening the Seguro cactus. I think question 3 was invasive species on the 2024 also. That's funny. 2 months on people? Man, that's tough. That's tough. Buffalo grass is an invasive grass species in southwestern desert ecosystems. It's threatening the Seguro cactus. Yeah, so this is we'll see that. We'll look at this. I think Lamarck stuff maybe. Yeah, I didn't really spend. I don't think I talked about Lamarck at all for my AP Bio class actually. So, because he's not on the test. It's threatening a Seguro cactus, a keystone species in these ecosystems. Buffalo grass is a drought-tolerant is drought tolerant and can survive wildfires. However, the dry buffalo grass also acts as fuel for wildfires, causing these fires to be more severe. Older saguaro cacti can survive wildfires, however, many of the young ones cannot. Scientists conducted an experiment to determine whether they could control the abundance of the buffalo grass population. The scientists identified several native grass species that, when grown with buffalo grass, might reduce the abundance of buffalo grass. They grew buffalo grass in the presence of different native grasses in the greenhouse in either non-drought, watered every 3 days, or drought, watered every 9 days conditions. After 12 weeks, they measured the height and dry weight of buffalo grass. And so the idea here is buffalo grass is an invasive species, and they've identified native grasses that can reduce the abundance of buffalo grass. Describe the effect of removing a keystone species on an ecosystem. This is the definition of a keystone species, right? Removing the keystone species will reduce stability in the ecosystem. There's probably a number of ways you could say that, so we'll just look at the way they say it. Reduces biodiversity, resilience, cause the ecosystem to collapse. You could even say something very general like this. Very simple idea. Identify the control group the scientists should include in their experiment. So this is a great question that you'll see sometimes, that there's not a control here, and so what would you do for the control? They have watered every 3 days, watered every 9 days, so they're changing drought versus non-drought. The control that I would add here is I would do buffalo grass without the native grass species, right? And that's what I would do. State the null hypothesis of the experiment in which buffalo grass is grown in the presence of native grass species. The null hypothesis is that the presence of native grass species will not affect the abundance of buffalo grass. So, scientists have found that the population growth rates of native grasses are much slower than population growth of buffalo grass following a wildfire. Scientists claim that wildfires are therefore increasing abundance of buffalo grass plants in the ecosystem. Based on the information, justify the scientists' claim. So the scientists claim that wildfires will therefore increase the abundance of buffalo grass, right? Because how do you justify that claim? And it's because buffalo grass regrows quickly after a wildfire. It's literally what it just said. So, right. The null hypothesis is always the idea that the independent variable has no effect on the dependent, right? There's no change in the experiment. Good. Question 4. 20 million years ago, the Caribbean Sea and the Pacific Ocean were connected, and waterways flowed freely between the two bodies of water. Many of the same marine species were found in both areas. Over millions of years, the land referred to as the Isthmus of Panama formed, eventually closing off the connection between the Caribbean and the Pacific Ocean and creating two separate bodies of water. The ecology of these two marine habitats was dramatically altered by this land formation. The warmer Caribbean Sea could no longer flow west, so the Caribbean water cooled and became more nutrient-rich. Describe the genetic evidence that evolution is occurring in a population. Love this. This is just the definition of evolution. There's a change in allele frequencies. That's it. Part B. Explain how the isolation of marine species by formation of a land barrier can lead to divergent evolution from these species. Divergent evolution is the same idea as adaptive radiation, and so isolation causes smaller population, right? And a smaller population is more likely to change quickly. We know that because, it won't be at equilibrium anymore, right? And it's going to be affected by things like genetic drift and that kind of thing. And it's going to be more likely to change. And there's going to be more available niches for them to do. The formation of the Isthmus of Panama connected two continents, North and South America. Many North American land animals migrated south after the formation of the isthmus and occupied similar niches as South American species. Predict the effect of the effect the formation of the isthmus had on resource availability for South American species. It decreased the resource availability. Justify my prediction because there was new species entering into ecosystems. May want you to say something more complicated than that, but no. Decreased, more species are competing for the same resource. It's just literally the same thing. I mean, guys, if you get this question exists, right? And so this is a perfect example of why you shouldn't fret about this test. Question 5. Figure 1 shows the reactions of a metabolic pathway used to synthesize amino acid B from amino acid A. And so we have A, and then you have this stuff going on, and then you have B. Describe a characteristic of an enzyme's active site that allows it to catalyze a specific chemical reaction. My students will love this one. It's because the shape of an enzyme determines its function. Or you could even be more specific and say the active site is specific to a substrate because its shape is specific. Based on Figure 1, explain how the binding of amino acid A to enzyme 1 is regulated by amino acid B. Explain how the binding of amino acid A to enzyme 1 is regulated by, okay, because the production of amino acid B activates enzyme 1, right? And so explain how the binding of amino acid A to enzyme 1 is regulated by B. The production of B is going to cause enzyme 1 to be activated, right? You could say that it becomes a inhibitor for that, and it's actually, I see that now. Look at this. I missed this at first. Okay, good. So, and this is the same shape. Enzyme or amino acid B becomes a competitive inhibitor, right? And yeah, it binds to the active site. Actually, you could argue non-competitive inhibitor because of the pictures are weird. I'm actually curious how they score this one here. Inhibits. You could just say inhibits the binding of amino acid A to this says allosteric site would be the non-competitive inhibitor, but you don't even have to be specific. You could just say that. Yeah. This, anytime you see this right here, this means inhibits, and so the production of amino acid B causes enzyme 1 to prohibit the binding of amino acid A to enzyme 1. That's it. So this would be like a negative feedback mechanism. Using the information in Figure 1, identify the product of the reaction catalyzed by enzyme 2. So enzyme 2, here's the substrate that binds to enzyme. Intermediate X is the substrate that binds to enzyme 2, and intermediate Y is the product. Right? And based on Figure 1, explain how a change in pH would affect enzyme 3 in such a way that enzyme, it's going to change the shape of enzyme 3, and that's going to change the active site. It's going to change its function. Whew. All right. The talking's getting to me. It's getting to me. I got this, though. We're going to do this, and then I'm going to go through photosynthesis. I got this. Yeah, you could use non-competitive on that one as well, and that'd be fine. Sometimes being specific is fine, but they also will say non-specific. And yeah, can we just say it denatures it? Yes, 100%. Denature is the word for changes its shape. So totally, you can always use your vocab, and it'll be right. Good. The ALD gene in fruit flies encodes the ALD protein. which is associated with both centromeres of chromosomes and protein filaments produced in meiosis. In the absence of the ALD proteins, gamete-producing cells enter anaphase 1 before homologous are correctly aligned. So if there's no ALD, then it screws up the cell cycle. As a result, gametes produced do not contain correct. Scientists generated 4 mutations in the ALD gene, ALD1, 3, 23, and del, which is a deletion of the gene. To study the role of the ALD protein in meiosis, scientists used gamete-forming metaphase cells from groups of flies with different ALD genotypes. Some of the flies were homozygous for wild type of ALD. Others were heterozygous for different ALD genes. Scientists measured the percent of metaphase cells that contained ALD-associated filaments and the amount of ALD protein produced. And here we have some of this stuff. Wild type has a percent of metaphase cells. Okay, so basically, as long as they have the ALD protein, whether it is, or as long as they have the wild type, which is going to be the ALD protein, these other ones are mutations, right? Then they are going to enter metaphase. These do not enter metaphase at all. Okay, and there's no difference between these two. So let's look. Based on Figure 1A, identify the fly genotype in which the average percent of metaphase cells with ALD-associated filaments is close to 12. That would be the ALD1 delete. Just this one. Based on Figure 1B, describe the difference in ALD protein production between gamete-forming metaphase cells, gamete-forming metaphase cells of flies with wild-type ALD323 and flies with genotype ALD323 delete. So this is electrophoresis here, right? The difference is with the ALD23 delete, there's no protein produced, and with ALD2323, there's a little bit of protein produced. That's the difference. Scientists hypothesize that gamete-forming metaphase cells can produce a normal amount of ALD-associated filaments, even when producing half as much of ALD protein as the wild type. Using the figures in Data 1 support the hypothesis. Yeah, so about half as much as the wild type produce. Okay, it's good. Wild type produces, as little as 40, and the ALD and this goes up to 20, right, which is about half, and you can see that here as well. That wild type produces a big block, and these blocks are about half, right? That's the idea. This one's kind of wonky. I'm actually curious what they say on this one. Yeah, shows no difference in the percent of the data for this and support the scientist hypothesis. Yeah, I don't know about that one. That one's kind of weird. I'm not proctoring the test. I'm going to be I'll be teaching my honors sections at that time. Actually, they don't let me proctor, so I'm glad. I would not want to proctor. I don't like proctoring tests. So for gamete-forming metaphase cells of this and this, explain why the phenotypes observed in Figure 1A differ than even though the amount of protein is not. Would I care if someone cheated on the AP exam? Yes, I would care. I don't even know how you would do that. I'm just going to move to this one. My brain is fried here. So let's see why the phenotypes differ because only produce high enough functional to generate wild type phenotype. Okay. So we're going to look at photosynthesis and cellular respiration now, as promised. And I'm going to go through this very quickly with you guys. So here's photosynthesis. There's my definitions of these things as well. That's great. And so here's what we're going to do. We're going to just track some electrons here, right? We're going to track some electrons, and that's what we're going to do. And that's how we're going to figure this out. No, we'll just do descriptions rather than tracking electrons. All right, so in photosynthesis, there's two reactions, light-dependent, light-independent reactions. Right. The purpose of the light-dependent reaction is to create these two molecules, NADPH and ATP. NADPH is an electron carrier. Those electrons come from water being split in the presence of light on photosystem 2. Oxygen is going to be produced as a byproduct of that process. ATP is produced on the electron transport chain in this because of the buildup of protons in the thylakoid membrane, right? Or inside the thylakoid. And so now we have ATP and NADPH made. Those two molecules are going to the stroma. So here's our light-dependent reaction. Right? Here's water being split. These electrons are going to go through the electron transport chain here. It's a series of redox reactions. These redox reactions are going to power the transport of protons from an area of low concentration to an area of high concentration inside the thylakoid. And so we have this concentration building up inside of here, right? Yep. Plants can do. Plants have mitochondria also. Don't forget that. And so those electrons are eventually going to be picked up here by NADPH, and they're going off to the Calvin cycle. And these protons are going to build up, and they're going to pass through ATP synthase, which is going to facilitate the production of ATP, which is also going to the Calvin cycle. Calvin cycle. Here's our Calvin cycle, which is also called carbon fixation. It's called carbon fixation because it's taking unusable carbon and making it usable. Calvin cycles, there's not a lot on the Calvin cycle. Cell cycles, I'm assuming like mitosis. Yeah, I could do that briefly. Yeah. So here's CO2 going in. Yeah, you need to know rubisco. It's just the enzyme that pulls CO2 in, like adds CO2 to the merry-go-round here. I call this a merry-go-round. Rubisco is probably the most abundant organic molecule on Earth. So there's your little trivia for today. So CO2 is added to the Calvin cycle, and it's 3 carbons that are put on. No, you do not need to memorize inputs and outputs at all. None. You just need to know ideas. Just ideas. These ATPs and NADPHs from the electron transport or from the light-dependent reaction are going to be used to create this G3P right here. And if you have 2 of those G3Ps, you will basically have a glucose molecule. And so we're going from, I think a picture like this is actually a whole lot more helpful than any of these reactions, because if you had this picture, you could answer most questions about photosynthesis. Almost most. Just a few things as far as AP bio concerned. Cellular respiration. Speaking of pictures that you need to answer questions, this is a great one, right? So here's cellular respiration. Where it shows the X and Y instead of the diagrams. I think I'll need a little more clarity on your question there. Can you describe where it shows it in the X and Y axis instead of diagrams? I don't know. Maybe I don't know what that one is. Maybe I don't think I have any of those. Oh, like this maybe? Is this what you're talking about? This maybe? Photosynthesis, yeah. It's wavelength, which is just color. This is absorption. This is showing you that green is reflected color, which is why you see green. Okay, so let's look at this. So cellular respiration is the opposite of photosynthesis. We're taking a glucose and we're breaking it down, and we're getting the products of cellular respiration, or the reactants of photosynthesis. It's pretty helpful. Yeah, I haven't seen that graph, the energy levels. I haven't seen that graph. The practice exams on AP Classroom are going to be pretty close. We use those in my class as well. They're pretty good. They're going to be pretty close. So. Yep. And those practice exams are like closed, and so they're only available on AP Classroom and probably on Quizlet. Sadly, yeah. If you really want to go look at some AP questions, I don't know. The Quizlet may have taken them all down now, but they used to all be on there, which was really sad because some derelict teachers post them online, and so that's why they end up being out there. So here's cellular respiration. Glycolysis breaks down glucose to make pyruvate. There's some ATP made in glycolysis, some electron carriers made in glycolysis also. 2023 FRQ, question 4. Okay, I'll go look at that one real quick before we end here. Yep, you're welcome. Those pyruvates are going to go to this part called pyruvate oxidation. It's kind of like an intermediate section where one of the carbons is lost and the other two carbons are put on the citric acid cycle, or the Krebs cycle. The purpose of the Krebs cycle is to make electron carriers, NADH, FADH2. Some ATP is made, but not very much. The business end of cellular respiration is the electron transport chain, or oxidative phosphorylation, which I will show you a better picture of. And so in this, electron carriers drop off their electrons, which is going to power these proton pumps. They're building a concentration of protons in the intermembrane space, which is going to be used by ATP synthase to make ATP. So here's citric acid cycle down here. And this is called chemiosmosis, the idea of this building up and using this concentration gradient to make ATP. One other thing I want to talk about, which I've seen a couple of questions about, fermentation. Fermentation happens when there's no oxygen. And the purpose of fermentation occurs so that glycolysis can continue. It regenerates NAD+, right? Glycolysis can't occur unless there's NAD+, right? And so in order to get NAD+, NADH has to be able to drop off its electrons. If there's no oxygen, it doesn't have a place to drop off its electrons, and so you have a buildup of NADH. So what fermentation does is it allows NADH to drop off its electrons to regenerate NAD+, which goes back into glucose, or glycolysis, right? So you're only making these 2 ATP over and over again, but for some organisms, that is pretty much all you need. Now, there was a request for the 2023 FRQ, question 4. Is it? No, this is 6. 5. This question? Let's look at it. I'm actually curious about this question. It's been a while since I've looked at this one. So I do have videos going through the 2023, 24, and 25 FRQs in more detail if you want to go watch those this evening. Non-cyclic electron flow and cyclic electron flow are 2 major pathways of the light-dependent reactions of photosynthesis. The non-cyclic electron flow, so non-cyclic just means that they end up in a different place than they start, right? It's a line as opposed to a circle. Electrons pass through photosystem 2, then components of the chloroplast electron transport chain, photosystem 1, finally at NADPH. The cyclic electron flow, electrons cycle through photosystem 1 and some components of the electron transport chain, which is what you see here in this particular figure. I believe there is a timer, yes. Describe the role of chlorophyll in photosystems, in the photosystems of plant cells. Chlorophyll absorbs light, which is going to be used, that energy is going to be used to break water to make more electrons. All right. Part B, based in figure 1, explain why an increase in the ratio of NADPH to NADP plus will cause an increase in the flow of electrons through the cyclic pathway. Let's look at this picture here. And so the cyclic pathway is here. Explain why an increase in the ratio of NADPH will cause an increase in the flow of electrons. As NADPH is being made, the energy that is the energy exchange here on this particular protein, ferredoxin, is going to be used to create extra ATP for the cyclic pathway. That's what you're seeing here. As NADPH is being built, so it's as one is being built, then it's going to change the ratio. Yeah, okay. Using rice plants, scientists examine the effect of a mutation that results in the loss of protein CRR6. CRR6 is the part of photosystem 1 complex, and its absence reduces the activity of photosystem 1, predict the effect of a mutation on the rate of biomass accumulation. If photosystem 1 is broken, electrons are not being passed through the electron transport chain, and therefore NADPH and ATP are not being made, therefore glucose is not being made. Do questions 1 and questions 2 with all my might. I don't have much might left over. I just don't. Sorry. I am waning here. Justify your prediction. I did that. So it would decrease biomass because it would decrease the amount of electrons made. Let's look at these. Okay, this is actually so yeah, let's look at this graph real quick. Just maybe some of you guys are a little bit concerned about the graph. I think this is a great example of a graph here. So construct an appropriately labeled graph that represents table 1. You've got 6 different species, right? And each one of those species is going to have mitochondria at normal CO2 and mitochondria at elevated CO2. And so in reality, you're going to have like 12 bars here. And This is a busy thing, but this is definitely a bar graph. Definitely a bar graph. Let's go up here and look at the prompt though, real quick. I may be wrong here. I think this may be a scatter plot. Scientists are studying mechanism to investigate. No, it's not a scatter plot. No, it's not, because these two are related to each other. So yeah, I can go over the steps of mitosis. I think one thing here to make sure that you don't include interphase in mitosis. The steps of the signal transduction pathway are reception, transduction, and response. Prophase is where chromosomes are still going to start being shown. Metaphase is where they line up the middle, which is here. Anaphase is where they separate. And then telophase is where you have still one cell, but the nucleus is separated during telophase. Okay. So I'm going to just end this evening with just a couple things to sort of just help you guys think about this. I think just a couple of hints. If I was in your shoes and I had to take this test tomorrow, what would I do? All right. So the first thing that I would do is I would realize that I have about a minute and a half per multiple choice question, and I would think about each question like that, right? And so when I saw a question, I have some practice questions on all of my. Let's see if we can find a really easy practice question. Man, some of my questions aren't very easy. Let's go. Let's maybe Unit 7 has some easy ones. Maybe not. All right, I'm wasting my time here. So you've all had multiple choice practice, and you realize that there's some questions that literally take you 10 seconds to answer. So if you have a question that takes you 10 seconds to answer, then do that. And now for your next question, you have 170 seconds, right? And so your multiple choice, you should not feel rushed in it. You should feel like it's a pretty good pace. As long as you aren't lingering on questions, you should be fine. My recommendations for the FRQ would be to, as soon as you get your test, I would just go ahead and start on question 1. Because it's a digital format, I'm a little bit wary of skipping around, but I think you can still do that. You guys are probably more comfortable with that than I would be. If I was doing the test, I would do my graph question last, and my reason for that is it's going to take the most amount of time, and you wouldn't want to sacrifice other points because of that, right? And so that's what I would do. Is I would do my graph question last. And if you get to one that you're struggling with, then maybe write a couple. The FRQs are on paper, at least the booklet is. But if you get to one that you're struggling, maybe write a few notes there and then come back to it later. But don't linger on something. Don't sacrifice the rest of the test for 1 point, right? Because literally every one of these words are 1 point each, right? 1, 1, 1. The only one that's worth more is the graph question, and it's only worth 3. So don't sacrifice the whole test because you're worried about this one calculation, for instance. Just don't do that, right? If you guys have specific content questions at this point, I encourage you to to go, well, there's a couple of options for you actually. I have review videos for each unit. Another thing I have, and this may interest some of you guys that you may not know if you have not. Let's see if I can pull this up here. I might be able to show you here. Let me see if I can. No, I don't think I can show you. So I'm on my YouTube channel here, and I have YouTube Shorts for every unit. And so if you just go to my channel page, the front page of my channel, there are YouTube Shorts for Unit 1, Unit 2, and they're like 30 seconds each. And just watch them. I'm going through it. You could watch all the shorts while you're laying there getting ready to go to bed, and it goes through all the content really quickly. So that's a recommendation. If you're wondering how to get some last minute content in, just watch those shorts. I think that would be helpful. Yeah, how in depth you need to know those things. It's not as in depth as you think. Trust me, and probably not as in depth as a lot of your teachers went into that kind of stuff. So I have confidence. Use your time wisely. If you have specific questions, you can put them in the comments, and I'll try to answer them as I see them this evening. But get a good night's sleep, and don't be late. Don't be late for the test. Thank you guys for watching. It's been a lot of fun. And let me know. And I'll be releasing the FRQ videos a couple of days. As soon as the FRQs go live, I will be making videos going through each of those FRQs. And so let me know in the comments how you think you did. And yeah, thank you.